Good morning, everyone. Once again, welcome to the Department of Pediatrics Grand Rounds. My name is Debbie Babcock. I'm a member of the Pediatric Grand Round Committee, and I'm a primary care pediatrician at Altos Pediatric Associates in Los Altos. We've been part of PCHA since 2018. Today's session is part of the Ann T. Bass lecture series in Hematology, Oncology, and Stem Cell Transplantation entitled Global Strategies for Sickle Cell Disease. It will be presented by Russell Ware, MD, PhD, who is the director of the Division of Hematology at Cincinnati Children's Hospital and professor in the University of Cincinnati Department of Pediatrics. But first, there are a few things to remind you of. I want to bring to your attention the text code for CME credit. It will also be put into the chat box if you don't get it right now. And we do have a number of exciting and interesting programs coming up that I want to remind you about. So first of all, our own Grand Rounds next Friday, April 8th. We welcome Dr. Howard Dubowitz from the University of Maryland School of Medicine. He'll be presenting on neglected children, a role for healthcare professionals. Then on Friday, April 18th, we will have the annual Rudine DiCarlo Memorial Lecture Series presentation given by Dr. Perry Klass from New York University, and she'll be speaking on One Vacant Chair, How Childhood Death Became Unnatural. But in addition, there are a few other things coming up to think about. Next Monday, April 4th, is the MCHRI seminar series with the topic Artificial Intelligence in Medicine and Imaging, Examples from Maternal and Child Health. Registration is open. Then on Tuesday, April 12th, is the 6th Annual Frontiers in Diabetes Research Symposium, and that will be at the Li Ka-shing Center, Berg Hall. So scan this QR code here for registration. You can see that there. Then we also have on Friday, April 29th, the 13th Annual Pediatrics Research Retreat at the Li Ka-shing Center, Berg Hall. And here's another QR code to scan for that registration. And then finally, the Pediatric Academic Society's meeting for 2022 will be held in Denver, Colorado from April 21st to 25th. And Dr. Fernando Mendoza, our own Dr. Mendoza, will be honored with the Joseph St. Jem Award, Leadership Award at the Department of Pediatrics reception on April 23rd for those of you who can be there. Okay, so now I want to hand off to Dr. Bert Glader, who will introduce our speaker for today. Dr. Glader is well known to all of us here at Stanford Children's for his leadership and contributions to the Division of Pediatric Hematology Oncology. His recent research and scholarship have been in the areas of pyruvate kinase deficiency and diamond blackfan anemia. He is the recipient of numerous honors and awards, not the least of which are several awards recognizing his outstanding teaching and dedication to the next generation of clinicians. So, Dr. Glader, thank you very much.
Thank you, Debbie. Good morning, everyone. It's a pleasure to be here. Wish we were all together in person. This is the Ann Bass annual lectureship in hematology, oncology, and stem cell transplantation. It's in honor of Ann Bass, who was a major benefactor of the Children's Hospital in developing the new hospital, fundraising. She was on the board of directors, and she and her husband together helped us establish the Bass Cancer and Blood Center. So it's in her honor that we have this lecture every year. And we split it every year, either goes to hematology, oncology, or stem cell transplant. And this year, we're fortunate to have it, be able to invite a hematology speaker. And the selection, when we discussed possible candidates, was absolutely unanimous. Dr. Russell Ware. Dr. Ware is a national and internationally acclaimed hematologist. He's long-term interest in sickle cell disease. He's published over 400 articles on these disorders. And just a bit of his background, he graduated medical school at Duke and pediatric training at Baylor, and then returned to Duke for his hematology oncology training, at which time he also got a PhD in immunology. Subsequently remaining on the faculty at Duke for several years, and then with stays also at St. Jude and Baylor, And then last 9 years, he's actually been in Cincinnati, where you heard his titles. He's the director of hematology there, and also the director of the global strategy program and the global health program at Cincinnati. Yesterday, we spent time talking with Dr. Ware about hydroxyurea, which is a major interest of his. But also, getting hydroxyurea out to the rest of the world is a real issue, and that's what we're going to hear about this morning. The title of his presentation is the Global Strategies for Sickle Cell Disease. Russell, welcome.
Thank you very much, Bert. What a kind introduction, and Dr. Babcock and the entire group for hosting me and honoring me with this lecture. It's a great privilege to speak to you today. I'm going to talk about my own story with sickle cell disease and global strategies, but I hope that some of the lessons will be potentially useful in terms of how your own careers are going, and if you can involve international health or in any form of global health, whether it's local or international. This first slide is important for a couple of reasons. It shows, of course, that I'm at Cincinnati Children's, which is supportive not only of hematology, but of global outreach. But the cartoon on the right shows the crushing burden of sickled cells on the child. And I think it's a wonderful representation of what we're only now in the last 10 years or so recognizing, that like HIV, like malaria, sickle cell has a really crushing burden of disease on children. And so I'm happy to describe it to you today. I do have some disclosures. I get some research donations from several groups. I'm on one medical advisory board, and I serve on the data safety monitoring board for several clinical trials. I'm going to divide the talk into a couple of parts. I'll begin by bringing everybody up to the same level of understanding about the burden of sickle cell by describing its pathophysiology, its manifestations and origins, and then spend the bulk of the time talking about current global efforts, some that we're doing, some that others are doing, and then end with a brief discussion of what would be some future strategies. Sickle cell has been around a long time. This is the first description of sickle cell in the medical literature, in the Western medical literature, and it was a publication in 1910. And you can see that the title actually has the word sickle in it. And so it was coined right at the beginning as a peculiar, elongated, and sickle-shaped red blood corpuscles in a case of severe anemia. Dr. Herrick was a cardiologist and was seeing a young man who came in with jaundice and heart palpitations and was weak, and he had anemia. And when he looked at this young man who was a dental student, he first understood that he came from Grenada in the Caribbean, and that he was a student in the Chicago area. And when he saw this patient, he realized that this was something different, and his intern, Dr. Irons, did a microscopy analysis and saw these shapes. And these are Dr. Irons' drawings of the blood cells down at the bottom. And Dr. Herrick described them as not only unusual blood findings, but freakish poikilocytosis, something only a hematologist could appreciate. But this cardiologist wrote about that. And academic medicine being as it is, he's a single author, and poor Dr. Irons didn't make it into the authorship list. So some things haven't changed too much. But this was 1910, so we're over a century of sickle cell anemia, and a lot has been learned. For example, we know very well, as sickle cell was probably the first molecular disease, that is due to a single point mutation in the beta globin gene. And this changes fundamentally the structure of hemoglobin. So this molecule at the top left is a hemoglobin molecule. It's got 4 protein chains, 2 alphas, 2 betas. Inserted in the protein chain is this little planar wafer called the heme ring that has iron and oxygen. But the sickle mutation isn't anywhere near where the alpha and beta touch, or where the heme ring inserts, or where the oxygen is. Instead, the sickle mutation is out here on the edge. It's right at the interface then between two hemoglobin molecules that normally are in solution, but because of the mutation, it allows one hemoglobin molecule to touch another. And very quickly, one hemoglobin touches another, touches another, touches another. And in the deoxygenated state, when the hemoglobin molecule has released the oxygen, the hemoglobin molecules that are normally in solution begin to contact each other and polymerize. And very quickly, they become these fibers, these rope-like three-dimensional structures, and that then stretches out the shape of the cell into the characteristic sickled form, which was coined presumably because of the farmer's sickle that has a curvilinear shape. And this is the essence of the pathophysiology, that in this cross-sectional analysis, you can have blood cells that are nice and round. They have a concave divot in the middle that is characteristic of the biconcave disc shape with oxygen. And as soon as the oxygen is removed, they become these distorted, elongated shapes because the hemoglobin is now polymerizing inside of them and stretching them out. And as the cell becomes reoxygenated, that polymer dissolves, but eventually the membrane is so damaged that it retains that shape and eventually breaks apart. So when I refer to the phrase sickle cell disease, I want to make sure that we understand some terminology. This term encompasses any blood disorder that the majority of the hemoglobin inside the red cell is sickle, what we call hemoglobin S. So this operational definition excludes sickle cell trait, because in sickle cell trait, you have about 30 to 40% hemoglobin S. And as a minority part of the hemoglobin milieu, it almost never can sickle in physiologic conditions. Say almost never. But in disorders where sickle is the majority of the hemoglobin, greater than 50%, often greater than 90%, these are what we would call the sickle cell disease. Now, at the genotypic level, patients can inherit hemoglobin S from each parent and get a double dose. This is the most common and the most severe, homozygous hemoglobin SS. But a child might inherit hemoglobin S from one parent with thalassemia. And if it's a beta 0 thalassemia, that is just as severe, and we call that sickle cell anemia. If it's S with C or S with another form of thalassemia or S with another variant, then they have different combinations. So sickle cell disease is not one entity. It often is interchanged with sickle cell anemia, but in our parlance, we prefer that the genotype be used. So a child might have hemoglobin SS or might have hemoglobin S beta plus thalassemia. So when we look at this entire group of disorders, we recognize several things. So in the United States, about 3 million persons have sickle cell trait, primarily African Americans, but now really in every patient population. And if you do the math, that means that about 100,000 Americans have sickle cell disease. But that's only a fraction of what the worldwide burden is. There are 400,000 annual births, and Africa, particularly sub-Saharan Africa, has the greatest burden. The clinical manifestations of sickle cell may be known to you. They're somewhat predictable in categories, but in any individual patient, they're very much not predictable. One common element is an increased susceptibility to infections. The spleen very early in life gets clogged up with these sickled cells. And so even though the spleen is anatomically there, it doesn't function in its proper filtration mode. And so we call this functional asplenia or functional hyposplenia. And those children are susceptible to bacteremia from pneumococcus and other encapsulated organisms, becoming full-blown sepsis and often fatal. And so functional asplenia is associated with invasive pneumococcal disease, which is why we use prophylactic penicillin and early and complete pneumococcal immunizations, including the polysaccharide Pneumovax. The acute vaso-occlusive events are what are most familiar for this disorder. Patients and families dread these. These are the acute onset of painful events. Clogging of the sickled cells in the lungs causes what we call the acute chest syndrome. It's often triggered by pneumonia, but it doesn't necessarily involve infection. Acute vasoocclusion of vessels in the brain causes stroke. It can trap in the blood. Blood can trap in the spleen, causing what we call an acute splenic sequestration with hypovolemia and shock. And priapism, which is a trapping of the blood within the penis. So this is a prolonged, unwanted erection. It can be very, very painful and damaging. I mentioned that red blood cells fall apart when they get stretched out. And so all patients with sickle cell disease have an anemia. It's partially compensated by making more reticulocytes in the bone marrow. But over time, breakdown of red blood cells, just like with any hemolytic anemia, causes jaundice, gallstones due to bilirubin, and even a release of hemoglobin and hemopexin in the blood that traps and depletes nitric oxide, and this leads to vascular disease. But what we know over time through natural history studies is that patients rarely die of the initial complications unless it's infection, but they do die of chronic organ damage. And over the years, even if they're not in an acute crisis, they have damage to many organs, really every organ, including the spleen, the kidneys, the brain, lungs, heart, bones, eyes. And so that's this inexorable progression of chronic organ damage in the second and third decade of life that is really the major cause of morbidity and mortality now, at least in the developed world. So I would like to impress upon you that multi-organ damage is bad and it leads to early mortality. We don't normally think of sickle cell disease as a fatal disease in the United States, but we should. Persons with sickle cell disease almost always will die of their sickle cell disease. It might be in their 20s or 30s or 40s. Worldwide, of course, it's very fatal at early ages, but we should realize and think and consider and treat sickle cell as a fatal disorder. It is not a benign condition by any means. So how did we get into this mess? Well, sickle cell as a trait has been identified genetically as arising in multiple parts of the world. Here is a diagram on the left that shows origins of the sickle mutation in West Africa on the far west, around Benin and Nigeria in the central region, and then the Bantu area in Middle Africa. There's also an Arab Indian. Now, this all is the same sickle mutation, and so these haplotypes, as we call them, are based on the flanking DNA sequences so that you can recognize where someone's sickle cell arose. And if you look at the heat map on the right, you can see that it really is concentrated in certain areas of sub-Saharan Africa, but for the most part, it goes across, and the population of Africa is, of course, so large, as well as India, that it really is a global problem. And through forced diaspora with the transatlantic slave trade, as well as more modern migration, there really is sickle cell disease in every corner of the world now. So what did this actually provide? Why would a deleterious mutation arise and not get snuffed out? Most deleterious things are not encouraged and don't propagate. Well, it turns out that hemoglobin S provides great protection against malaria. So sickle cell trait arose and was propagated because it confers protection against malaria, which, as you know, is also a killer of children and adults. And in fact, if you look on the right, the heat map of sickle almost exactly overlaps with the heat map of malaria, almost like a hand in glove match. And so what is presumed to have happened is that malaria is such a lethal condition that any spontaneous mutation that conferred a survival advantage, that child would grow up and be able to have children. And so as hemoglobin S arose in different parts of the continent, it actually conferred survival and was allowed to propagate and continue. In fact, most red blood cell disorders that hematologists take care of, whether it's G6PD deficiency or elliptocytosis or sickle cell disease, alpha thalassemia, all of these arose as a means of escaping the fatal malaria. And so I guess in many ways, hematologists owe a debt to the Anopheles mosquito, which is the transmitter of that. But it's an interesting story because if you think about it, then hemoglobin S as a single gene mutation is advantageous to have, but a double dose is fatal. And it is that what we call a genetic, mixed genetic polymorphism, where you get a benefit from one copy and damage from two, balanced genetic polymorphism. So now what about diagnostic methods? If you break open red blood cells, the majority of the protein is hemoglobin. And so it's relatively easy to separate those hemoglobins by gel electrophoresis. This has been known for 75 years. And in fact, sickle cell anemia can be diagnosed based on the location of common hemoglobins. Hemoglobin A is adult normal hemoglobin, S is sickle. There's a common variant from West Africa called hemoglobin C. And then all of us have hemoglobin F, fetal hemoglobin. And so depending on when you test, whether you're testing a child or an adult, they have different amounts of hemoglobin F. And on the left side is what we call a cellulose acetate gel that separates hemoglobins. The next one, stained in brown, is what we call isoelectric focusing that separates these. You can separate proteins by high performance liquid chromatography, and you can even do DNA-based testing. And all of these diagnostic methods are now well established and quite available, which is important because newborn screening requires some accurate methodology. Now, I can tell you in the year 2022 that the United States does sickle cell screening very well. We started in the 70s and 80s with targeted screening, limited screening. Certain states came on board, adding it to their newborn screening panel. It took until 2006 for all 50 states, the District of Columbia, and US territories to screen for sickle cell. So we now finally have universal newborn screening. You may have thought that this was done a long time ago, but it's relatively recent. And about 4 million births per year occur in the United States. They're tested according to the state's own panel of newborn screening. Usually it's done by isoelectric focusing. A few states do HPLC and a couple of states do DNA-based. And depending on the year, 2,000, maybe 2,500 new cases are identified a year. But this next factoid is true, and it's important to remember that although we talk about newborn screening in a variety of ways for genetic and metabolic disorders, hypothyroid, galactosemia, sickle cell disease is the most common disorder identified by newborn screening in the United States. And the stamp on the left honors the time that all the states were finally coming on board for universal screening. What do we do with newborn screening? There's no point in screening if you don't have something to offer. Well, we offer early education, we begin penicillin prophylaxis, and we make sure that pneumococcal immunizations are caught up. We also begin transcranial Doppler screening for stroke risk. And after that, we have access to therapy. We have blood transfusions, we have hydroxyurea that I'll talk about. Eventually, we have transplantation and even newer medicines that have just been FDA approved. And based on this, the NIH has published evidence-based guidelines that talk about the importance of screening, intervention, and early therapy. It's true that simple interventions save lives. On the left are survival curves in different eras, different parts of the world. And you can see that at the top in purple is our current survival. We fully expect children born in the United States to survive to adulthood. There might be 95, 98% survival, but it's essentially complete. However, it wasn't always that way. And these other survival curves are in the 1970s and 80s. And this is due simply to screening and starting penicillin prophylaxis and pneumococcal immunizations that has brought it up. On the right, you can see a further inflection with treatment. And so with the advent of hydroxyurea and safer, more available transfusions, we now have life expectancy that's over age 40. That was identified a couple of decades ago. It's recently been reviewed and found that it's still only in the mid-40s. So it does appear that unless we do something different, such as beginning disease-modifying therapy early in life. It's the chronic organ damage that will eventually lead to early mortality for these patients. I'm going to shift now to talk more about the global burden. I mentioned 400,000 births per year. If you do the math on how many cases I said occur in the United States, if it's 2,000, that's less than 1% of the worldwide cases. So even though we do most of the investigation, we do the research, we think we know a lot about sickle cell, it is a pittance compared to what the worldwide burden is. So even though we do newborn screening in the US and the UK, it's limited in other parts of the world and very limited in the highest burden districts. In Africa, we think that 10 to 25% of persons have sickle cell trait, which means at least 1% of the babies have sickle cell disease. That's an incredibly high rate of abnormal findings. Most of them die at age 5 or 10. It was often reported by survival analysis that at least 50 to 90, even as high as 90% of children died with sickle cell before reaching adulthood. Two very good epidemiologic studies have recently shown that about a third of them will die by age 10. So even in a more modern era in Africa where there's better diagnostic and management, still a very high death rate. And these deaths occur from infection and pneumonia and malaria because in limited resource settings, there's minimal access to diagnostic testing, vaccines, transfusions, and hydroxyurea. And if it's bad now, it's only going to get worse because of the birth rate explosion in Nigeria, Democratic Republic of Congo, and India. This cartogram shows the swollen and disordered map of Africa and India over the next generation. And it's been predicted that by year 2050, there will be 30% more new cases of sickle cell disease. So because of the existing malaria pressure, sickle cell trait is not going to go away. So unless there's efforts to identify persons of childbearing years having trait and consider education before having children, which is has its own set of sociological issues, we're going to have even more sickle cell disease in the future. The WHO has taken note of this and has issued 2 papers, one in 2006, one in 2010, and it's pretty bleak. This is from the 2010 white paper. Current national policies and plans are inadequate. Deaths occur mostly under age 5 years, and this would be from a variety of reasons, including infection, as well as splenic sequestration, acute chest, and so on. But in addition to writing this bleak report, I want to show you that it's not even recognized by everybody. Lancet every 10 years puts out a tally of child deaths, and here's the one from 2012, so we're awaiting one hopefully come out this year. And these are the cause of deaths. They do it for every continent in the United States, in the world. And this is the one for Africa, the pie chart of deaths. And you can see there's a lot of deaths from pneumonia, malaria, but sickle cell disease isn't even listed. You have tetanus, you have measles, but you don't even have sickle cell mentioned in Africa in 2012. And for us, and many, we view this as a question of equity. Why should this child on the left who has immigrated to the United States get access to early diagnosis and treatment with hydroxyurea? And the little baby on the right from Angola, who was part of our newborn screening program there, doesn't have access to any of that. His mother loves him just as much, but for us, it was a question of equity to try to take what we've learned from the US and export it. And we had guidance from WHO because at the end of their 2010 document, they gave some instructions. They said, here are the targets that ought to be done, surveillance, management, national plans, and they issued guiding principles about how to do it. And these are really important to sort of get away from the colonialistic approach of international health that has happened for several generations, and even priority interventions. So this was a time in my own career that I was interested in pursuing this, and we decided to focus on these areas, that we would first identify partners and work on surveillance, help build capacity, But if you look at the last one, research promotion, this might seem a little strange. After all, these are low resource sites. Are they really going to be able to do research? But I like this inclusion very much because my own training involved research as well as clinical care. And it's my firm belief that if you find good partners, help train them, you can conduct high quality research in Africa and in Caribbean and other low resource settings. The findings are even more important when they're conducted in these settings because they involve local patients, local clinical research teams. The results are generalizable across countries in that area, and they're better believed. And so we think that's an important part. And I'll show you how sparse the research is in Africa and around the world. This is a map that comes from the website called clinicaltrials.gov. I looked yesterday and there are 409,000 trials that have been registered in clinicaltrials.gov across 220 countries. That's a massive database of clinical research. When I looked to see how many have sickle cell disease and hydroxyurea, there were 51 trials. And you can see that the countries included, 41 of them are in the US, and a few of them have multiple sites around the world, 6 in the Caribbean and 9 in Africa. Considering the burden where there's over 100 times more sickle cell in Africa than in the US, that's a little bit startling. So over the last 10 years, my team at Cincinnati Children's has really spent effort expanding the international arena of research. And we've focused in 3 areas: diagnosis, surveillance, and treatment. And we now have active studies in 2 Caribbean countries, as well as 7 in sub-Saharan Africa. I want to first talk about diagnosis. I told you how easy it was to do isoelectric focusing in HPLC, but that takes technology, that takes electricity, that takes training of technicians. If we can find out in 5 minutes if you have COVID, if you are pregnant, if we can identify somebody by a blood test or a urine test for all of these things, why not sickle cell disease? And there are these rapid diagnostic devices that are being created by several companies with the support of NIH, a small business initiative, to create what we call point of care sickle cell diagnosis. And I think this is pretty good. We've published with at least a couple of these devices. They're not great. They don't have a way to identify whether you've got a little bit of hemoglobin A or a lot of hemoglobin A. But depending on how you do it and in what setting, you can actually get pretty clear distinction between normal and disease. There's also a group at Georgia Tech led by Wilbur Lamb that is trying to do point of care colorimetric analysis of hemoglobin just to know how anemic someone is. And he's developed a really clever device that actually kind of is based on the old Guaiac test that involves colorimetric change, and he's able to slide the scale to where you can very easily get a diagnosis of severe anemia under 5 if it's kind of a blue-green color, or if it's less severe in the orange to red color. So these are possible additions to the armamentarium as we go forward, and these are of course valuable if you don't need to have electricity and specialized equipment. The first surveillance study that I got involved with was in the Republic of Angola. And I show you the map of Africa because most people, including me, didn't know where Angola was. And I had the opportunity there of all kind of unusual funding sources from the Chevron Corporation. They and many other US and global companies get a lot of oil from Africa, a lot of oil from Angola. And as part of their corporate social responsibility, they wanted to help Angola, and they asked the Ministry of Health, what can we do? And they said, help us build a sickle cell program. And they said, right, we'll do it. What is sickle cell? And then they came back and I got involved with it and decided to make this an important part of my career. So when we got there, we learned a lot. The population, it's an enormous country, but it only has 20 million people because it had had a 25-year civil war. It was estimated that 10 to 20% might, as high as 20, might even have sickle cell trait. That would mean 5,000 to 10,000 babies born every year. no newborn screening, and a high under 5 mortality. So we decided to do, as I described, we helped build capacity, we trained, we brought people over, we did dried blood spots just for sickle cell. It was the first newborn screening that they had ever done. And they're, of course, very good at it. It just requires training. And in our first reports, we found a massive burden of sickle cell. So here's a report of 36,000 tests done. The fetal is listed in all of them because these are newborn babies. You can see that the number that have sickle cell trait, FAS, is almost 8,000. FS, which is disease, is 550. I'll put that in perspective. If in the US we had tested 36,000 samples, we would have somewhere between 12 and 14 babies to be identified with sickle cell disease. This was 550. Imagine that coming to your clinic for care. It was just staggering. And you can see the pie chart of how much trait and disease. We were able to find most of the half of the babies. We brought them into care. And it was interesting that they actually had lower mortality rate than the average baby or the baby without sickle cell disease in Angola. And we did cost-effective analysis as well. We took that to then say, well, maybe we can work with other countries. And here's Charles Chiaga, who runs the HIV lab in Uganda, who said, yes, let's do sickle cell too. This was a funny kind of coincidence because they had just started national surveillance of HIV, trying to identify mothers who were born, who were giving birth to babies to see if they could prevent the vertical transmission. And so they were collecting these early infant diagnostic EID blood spots across the country and bringing them in. And all we had to do was repurpose them and teach them how to do isoelectric focusing. So over a 12-month period, we were able to get about 100,000 samples from all over the country, show them how to do trait and disease. And we were able to create the first national geospatial map of sickle cell. You can see here on the left, this is the heat map or the color-coded map. And in fact, there's a lot of sickle cell in the country, but it's not evenly distributed. So the southwest corner is very light. The eastern part is lighter, but there's definitely a corridor toward the middle, which is very populated, that has a lot of disease. And so in some of these districts, you can see the ones in dark red and black, it's over 20% sickle cell trait. So this is just like I was describing in the area of Angola. We also found a lot of hemoglobin variants. That's kind of a scientific interest. And you can see a lot of different ones that are listed there on the right, different colors. We then took this to a neighboring country. Tanzania is just south of Lake Victoria, so south of Uganda. And they thought that they had about 10,000 babies in the whole country born every year. We were able to work with a team at the north called Mwanza Bugando Medical Center. We tested 17,000 dried blood spots, and we found, again, a huge burden. And this at least doubled the entire burden of the country. So this happened both in Uganda and Tanzania, that the estimates had to be doubled for the number of births that were happening every year. We then took it over to neighboring Malawi and worked with the UNC project there in Kamuzu Children's Hospital. There we didn't find as much sickle cell trait, but we found a lot of alpha thalassemia and G6PD deficiency. So it's kind of an interesting epidemiology that the protection from malaria may not be identical across sub-Saharan Africa. It may be that different red cell traits occurred in different areas. So now we have a different problem. We're telling Uganda that you have 2 or 3 times the amount of sickle cell that you thought. What are you going to do about it? And so there we needed to talk about therapeutic opportunities, because if you think about it from the Ministry of Health point of view, it's really not a help to them to learn that they have 3 times the burden that they thought. So that's just more children that they're not taking care of. And so we needed to provide some sort of linkage between screening and treatment. So here's a diagram from what we would say in the US is the cascade of pathophysiology of sickle cell disease. There's a lot of different steps that happen from red cell hemoglobin S polymerization all the way to organ damage. But if you can focus right at the top and intervene, then this would be the best. And this is where hydroxyurea comes into play. It goes right at the top of that cascade. It's a chemical that's been around for 150 years. It was used 2 generations ago to treat leukemia, to treat HIV until better medicines came around. There's a tremendous amount of experience with it, but it doesn't work as chemotherapy for sickle cell. It boosts fetal hemoglobin. So it prevents polymerization right at the top of that cascade by boosting fetal hemoglobin. It actually prevents it from being turned off after the babies are born. And so this is a fascinating collateral effect, if you will, of hydroxyurea. So even though we sometimes hear about it as a cancer drug or, oh, it's chemotherapy, it isn't. It's being used for boosting fetal hemoglobin. Guess what? Prednisone is a chemotherapy too. It's used in ALL treatment. But when you give it to somebody with asthma or poison ivy, you don't say, watch out, I'm giving you chemotherapy, because it has a completely different mechanism of action. And we should use the same argument for hydroxyurea. It's not being used as chemotherapy. It's being used to boost fetal hemoglobin. And it's FDA approved in the US and EMA approved in Europe for the treatment of sickle cell anemia. It works in a variety of ways. It boosts fetal hemoglobin, but it has a myriad of effects. It's really a remarkable drug. And even though we now have more drugs that are specific for certain parts of that cascade, we're unlikely to find one that does so many things at one time. Hematologists like to look at blood under the microscope, and on the right, you can see a before and after. This is after several months of taking it. You have many more blood cells. They're rounder, they're fuller of hemoglobin. It's not a cure. There's still some abnormal shapes there, but it's much healthier, and there's more blood. They're less anemic. It's been around for so many years that this timeline is pretty full, shows that the initial studies were done in the 1980s and 90s. We've done a variety of tests. The ones listed in trials, the ones in green are what we call phase 1-2 trials that are really early trials based on age. The ones in red are phase 3 trials that are testing it against standard care. So there's a lot of data on hydroxyurea, and collectively, it's very clear that it's safe and effective in all ages. The WHO includes it now in their list of essential medicines for children. It comes out every 2 years, and starting in 2011, it was listed as important for hemoglobinopathies, and specifically sickle cell disease. So when we began our treatment trials, we tried to meet countries where they needed it. And so we first went into the Caribbean, where they already were doing good diagnostic tests, but they said, we need something for stroke. We're not able to intervene, and we're having so many children have a stroke. So we brought in transcranial Doppler screening. This is something that's done routinely in the US. It's an ultrasound device. You can see it's just held against the temporal part of the brain. We trained people in these different countries and have provided not only the screening tools, but the hydroxyurea to treat. These have been very successful, and they go by different names. Extend was just published. Sacred and Sphere are going to be hopefully published later this year. But these are great ways to build capacity and help the children, of course, and links screening and treatment. I want to talk about 2 bigger trials, because these are ones that fall into that group that I mentioned earlier of actually conducting them in the country and trying to prove that the drug would work in a place that is low resource. So the first is called No Harm, novel use of hydroxyurea in an African region with malaria. So why that name? Well, it turns out that hydroxyurea has so many effects that one of them is on the endothelium of the blood vessel. And it was suggested by a group of French investigators 15 years ago that some of the effects might be harmful, because it would maybe help sickle cell disease, but make the blood vessel more susceptible to malaria contact. And so with this concern, there was reluctance in some parts of Africa to proceed with hydroxyurea, because there would be worse malaria or more malaria. And so in this trial, Chandy John at Indiana University, a malaria expert, and Robert Opoka, who's at the Makerere University in Uganda, we worked together to create a trial that would test this directly. And it was funded by the Doris Duke Charitable Foundation. It was a phase 3 trial where the primary endpoint was malaria. We were going to use hydroxyurea or placebo and count the number of malaria events, and used a fixed dose of 20 milligrams per kilogram per day. I show you on the right the projected enrollment. This is standard for many sickle cell trials, but especially in Africa. You have a projected enrollment and you could beat it any month, any week. The enrollment is so brisk because there's so many patients and they're so desperate for treatments that they're very eager to participate in research. So don't ever believe this comment that sickle cell patients or persons of low socioeconomic status are not interested in research. I just don't believe that to be true. And what we found in the no harm trial was that there was no increased risk of malaria, which was good. There were all the treatment benefits and not very many toxicities. So we considered this a very successful trial, and it was conducted in Uganda and published and provides good generalizable findings. The treatment effects are almost exactly what we would have seen in the United States. There's things that go up, but we wanted to go up. The hemoglobin, the fetal hemoglobin doubled, more than doubled. The inflammation and the reticulocytosis went down. This was really just what we wanted to show. It was not only safe, but it was feasible and effective. And the dose-limiting toxicities were almost equivalent, which again spoke to the safety of the drug. Here are some slides that show the different clinical indications. Some were more significant than others, but the most important part was that there was no increased risk of malaria. And if anything, there was more malaria in the placebo trial, and that became important in a second trial. And that is called REACH. And REACH is slightly different. REACH does not include a placebo. It is an open label trial where all the children who enroll get hydroxyurea. And it was done in 4 centers that you can see here on the map, begin in Angola, Democratic Republic of Congo, Uganda, and Kenya. And these are the investigators that are leading this. And this was done with a huge drug donation as well. And the goal here was to just see, could we enroll children and follow them over time, and would it be safe and effective? The goal was to enroll 600 children across 4 sites at a fairly young age. And the enrollment was again almost picture perfect as we matched the actual enrollment to the projections, and you can see the distribution of ages across the span. And the findings were dramatic. Not only was the amount of sickle cell decreased, vaso-occlusive pain and acute chest, but so was malaria, so were transfusions, and so was death. Dramatic, unexpected findings. And this led to my colleague Leon Shalolo presenting this as a plenary session at the American Society of Hematology, and then publishing it in the New England Journal. But I want to go back to the no harm trial, because when we finished the placebo arm, all the children were on this fixed dose. And you could see here on the right that very quickly, even if they had been on placebo, they could get back on track. And we decided to do a second trial where we would vary the dose. There's been a lot of discussion in our literature about 20 milligrams or 30 milligrams. And we predicted that a higher dose might be more effective, but it might be more toxic. And what we found was that wasn't true. We found that when we did that separation, in fact, the benefits were huge, but the dose-limiting toxicities were equal. And the Data Safety Monitoring Board halted the study early, took the children who were on the lower dose and said they needed to be on the higher dose. And this was also published, and Dr. John is the first author of this. So again, high quality research done in low resource settings with great partnership from the local teams. So in summary, I hope I've convinced you that sickle cell anemia causes a large global burden of disease. Our lessons from the US about screening, intervention, education, treatments can be applied. Screening and surveillance are realistic goals, and one hope is that there will be more newborn screening instead of just pilot trials, actually have more national programs. Definitely believe that clinical trials can be safely conducted and should be conducted in low resource settings, and that hydroxyurea is probably the only effective treatment right now that makes sense from a financial and a safety point of view. Future strategies, well, we learned that hydroxyurea reduces malaria, but we don't know how. We learned that it reduces transfusion need. It causes associated with less death. So we need to understand that. We need to have genomics and biomarkers. We have other kinds of sickle cell disease that need to be investigated. So the future is quite bright for new clinical trials. But then once you find something that's important, there's a need to implement it. And this implementation science is very important, not only in the US, but in Africa especially. How can we help kids get on the right dose? Can we actually personalize the dose and have them need fewer lab monitoring tests? And then, of course, we want to get the attention of global funding agencies, not just WHO, but USAID, Global Fund, Gates Foundation, et cetera. So this is all a work in progress. And with that, I'll thank all the people that work with us around the world. It's been a really fun part of my career to develop this global opportunities and have partners around the world. Thank you for your attention, and I'm happy to answer any questions.
Thank you, Russell. That was absolutely terrific. And you're getting a lot of chat in here about people congratulating you on your research and the presentation. So thank you very much. One question that comes up is, why was sickle cell disease not mentioned in the Lancet article about the deaths in Africa?
I think that the reason, the way these groups gather the data, it's based on what the country provides. And if they're not doing any diagnostics, then they don't know. It was very similar 30 years ago to HIV deaths weren't listed. They were being called diarrhea or infection. But once diagnostic testing became available, then they knew what it was from. I think these sickle cell deaths are lumped into the pneumonia and other causes of death. But if we now have point of care devices for screening or newborn screening, then it'll get rectified.
Another interesting question comes in about the hydroxyurea being donated in all the studies. What happens with transfusions after you complete the studies? What happens then after the study is over?
I think you mean what happens to the treatment with hydroxyurea. Yeah, so this has been a part of the negotiation with the ethics boards as well as the Ministry of Health. We have asked these companies to continue to donate it long-term and to follow this cohort of children over time, and they've all agreed. So we're not abandoning them. In the case of the Caribbean, they were actually able to use the results of the Extend trial to go to the ministry and say, you need to provide this, and they're providing it at pennies per capsule. So I think the goal is to not pull the rug out, because that's an absolutely important comment to make, but to continue it. What we still have are the haves and the have-nots, right? We have some children that are on it that continue to benefit and others that don't. And that's sad, but I don't really know how to get around that right now. Hydroxyurea is very inexpensive. That's the other part that ought to be mentioned. It's pennies per capsule. And so I think that it lends itself to wider implementation if it can just sort of be made and be brought into countries.
I think we talked about Gates foundations and people like this aren't interested or aren't involved, or.
Well, I'm not sure that that kind of advocacy is what I'm the best at, but there's a lot of people that are better for that. And pharma companies have a responsibility too, particularly the ones that are trying to make very expensive medicines. They should donate or be a part of this too. One proposal from a colleague from Nigeria has said for every patient who gets treated with a drug that costs $100,000, they ought to put 1,000 of that into hydroxyurea for low resource settings. That seems like a pretty fair ratio to me.
I agree.
All right, I see a few questions if I could answer it. The first one's a very good one about what percentage of under 5 mortality is due to sickle cell. That's been estimated by WHO to be as high as 16% of the under 5 mortality due to 1% of the births with sickle cell. So very much over proportion. And they reduced malaria. So that's a very good question too. How would hydroxyurea reduce malaria? Well, it turns out that hydroxyurea does have cytotoxic effects. That's why it was discovered to be effective against HIV and was a part of the early HIV regimens until highly active retroviral therapy came along. So it may have a direct effect. It also is an iron chelator, but there's also a benefit in myelosuppression. By reducing the white count, you reduce a lot of inflammation, and it may be that it actually allows the malaria, it prevents the malaria from adhering to the endothelium as quickly as it might otherwise. But that's definitely an important area of investigation for us. The next comment was, how much is the underestimation of disease impact because it's people of African descent? Oh my gosh, you've hit it on the head. This is exactly why it's so neglected. It's exactly why funding for sickle cell research in the US has lagged behind other diseases that affect primarily white patients. There have been papers published about the disparity of sickle cell funding versus cystic fibrosis funding, for example. So it's true and it's pervasive. And at the national and international level, even more. So this is one way of bringing attention to it. Toxicities of hydroxyurea. It actually turns out that there's very few bad toxicities to hydroxyurea. The main thing is you have to titrate it for your blood count so that you don't get it too high, sort of like a lot of therapeutic drugs. So I would maybe refer you to a couple of review articles, be happy to share it, but it's the doses that patients take are between 15 and 30 milligrams per kilogram, so a fairly narrow therapeutic window. CRISPR gene editing. Well, so I think that the cure of sickle cell disease is a hot topic, and NIH has decided to put $100 million and convince the Gates Foundation to put $100 million toward curing diseases, and the two that they listed are HIV and sickle cell, and they want to do them in Africa. And how you could cure sickle cell disease with CRISPR technology when you haven't even diagnosed them, I'm not sure, or how you're going to keep them alive until they can get it. But that's a big aspirational goal. I think ultimately you can manage a lot of diseases, but it would be great to have a cure. And there's investigators here that are working on it, and I wish them the best. Happy to help them too.
Russell, I have one final question. Why is sickle trait protective against malaria?
That's a really good question. All the red cell mutations confer some protection, but sickle cell trait is the most potent of all. It's more potent than thalassemia trait, than G6PD deficiency, than spiro and elliptocytosis. It does a couple of things. The first is that the malaria parasite goes inside the red cell and chews up hemoglobin, and it doesn't like sickle hemoglobin as much as hemoglobin A. So it's a taste test, presumably, the rate of degradation. But probably the most important thing is that it consumes oxygen inside the red cell. And when the oxygen level goes to 0 inside a red cell, even with trait, it will sickle. And so that cell starts to sickle and the spleen plucks it out. So sickle cell trait is protective when you have a functioning spleen, and you need both of those together.
Thank you. Well, again, Russell, thank you very much. This is absolutely a fantastic presentation. Lots of kudos to you.
Thank you very much, Bert. And thank you all for your attention. It's my pleasure and honor to be here today.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.